Okay, so morning everybody, and welcome to our strategic cereal farm in Scotland. I'm currently in a very snowbound East Lothian, just about a foot of snow in there, so that's a nice interesting one for this morning. I'm sure everybody else has got the same struggles dealing with things out on the ground. So today we're joined by David Aglen, the host at Balbirnie Home Farms. We've also got Fiona Giri, who's overseeing the project from AHDB's point of view, and we've got Fiona Burnett from SAC or SRUC, who's the trials coordinator there, and we've got Eric Anderson and Joel Williams as well with us. So we're going to give you an overview of what's going on at the strategic cereal farm, and then Eric and Joel are going to appraise our trials and create some background discussion for us there. So today, just moving on a slide, if you can, basis and enroso points, as per usual, if you can type them into the chat. So if you put your name and your postcode and your basis number in there, please, and then a little gap in your enroso number, and we'll take a note of them and we'll pass them on to the girls at basis. Today it'll be recorded and it will be put out on the AHDB website. Any questions we do receive, we'll try and get through them as best we can. I know there's going to be a few. I've certainly got a few already before we start, so there'll be quite a lot of questions, and we will actually put them around the panelists and put them on the website with the answers as best we can once we get the recording and put it live. So you can see we're on Twitter there, AHDB Cereals. Sheila does a cracking job, so you can interact with us. There's some cracking videos that David and the team at Balbirnie have helped us with to get things off the ground. A very difficult period. We had hoped we would have had an open day in July at Balbirnie, but that wasn't to be during COVID. And this is probably the best we can come up with without being able to get on farm. So we have a couple of trials kicking off this year, which we're all looking forward to try and beat the experts, so to speak, as we're looking at, and some of the work that David's been doing at Balbirnie over the last 10 years. So I think we're going to Fiona next, just to introduce a bit more about the project.
Good morning, I'm Fiona Giri and I work in knowledge transfer at AHDB. And I'm just going to quickly introduce the strategic farm programme, so what it is, why we do it, and how you can make the most out of strategic farm network. So we can go on to the first slide, please. So firstly, what is a strategic farm? A strategic farm is a 6-year project focused on driving innovation and productivity by putting research into practice through on-farm trials. And
sorry to jump in briefly. The audio keeps cutting out a little bit. You might want to speak a little bit closer to the thing, but sorry, carry on.
I'll come in, zoom in. So the strategic farm is a 6-year project, and we demonstrate research in practice through on-farm trials and demonstration. And this differs from a monitor farm, which is a 3-year farmer-led, farmer-driven initiative, which focuses more on personal, business and technical development. So in the cereals and oilseeds sector, we currently have 20 monitor farms, and David joins as the 3rd strategic cereal farm host. Moving on to the next slide, please. So David joins Brian Barker, who is the Strategic Cereal Farm East host, and he is in his 4th year of the programme, and Rob Fox, who hosts the Strategic Cereal Farm West, and he's in his 3rd year. So information is shared across the Strategic Farm network to enhance the learnings and to get the most out of the research findings. There are some common themes across the strategic farms at the moment, and those are looking to reduce inputs while maintaining productivity, and both are looking at using flowering strips to encourage beneficials and natural enemies. Also in the east, areas of interest include using cover crops to improve soil health and water quality, as well as looking to boost early crop biomass to improve yield. And in the west, they've also been looking at utilising varietal resistance to alter fungicide strategies, and looking at cultivation systems, cover crops, catch crops to improve soil health and crop rooting. So those are just a few of the things being looked at the strategic farms, and more details of the work going on as well as results from previous trials, can be found on their respective web pages, which are shown at the left-hand side of the slide. So moving on to the next slide, please. So what are we hoping to achieve at the Strategic Cereal Farm in Scotland? The vision is to bring research and practical farming together to work towards farming with both economic and environmental resilience. So we want to achieve this by using data to support decisions, and the focus will be to determine viable options for a profitable business, as well as helping give farmers confidence in making changes. Next slide, please. So behind the Strategic Cereal Farm Scotland is an extensive steering group who bring in lots of areas of work and expertise. So the steering group will come up with trial ideas and steer the direction of the strategic farm over the next 6 years. And by bringing together intelligence from across the industry, it allows collaboration and us to pull together a lot of knowledge to generate trial ideas that will positively impact the industry. And David will also share his thoughts and vision for the next 6 years later on in the webinar. Next slide, please. So finally, how can you benefit? Practical messages are at the heart of the Strategic Farm programme. All of the trials are subject to full cost benefit analysis, which is released alongside the results to show the economic impact, as well as other measurables which are gathered as part of the trials or assessments. And it's important that the farm hosts also share their experience alongside the data. They can bring in another element to the practicality of the work. How-to guides are also often produced to accompany the trials so that other people can do assessments on their own farms, and Farm Bench can be used to help cost out the impact of the trials or assessments. And your experiences are important, and sharing information or having discussions on Twitter or events is a great way to get involved and enhance the work that's already being done on the strategic farms. So I'd like to encourage you to please get involved. You can even be feeding in your ideas of things you'd like to see on the strategic farms in the chat box as we go through the rest of the webinar. So thank you. I'll hand back over to Chris.
Thanks very much, Fiona. That's a cracking introduction to what we're trying to do. So again, for those, we can see a few more have just joined. So if you do pop your basis and Enroso number into the chat function on the right, we'll get that logged and away with them. So now we're going to introduce David Aglen, and he's going to take us through his vision for the strategic farm and where he's been and where he's gone. Okay, David.
Good morning, everybody. Hope you can see me. Slight internet connection issues here, but welcome to Fife, such as we can get a visit these days. I'm just a quick introduction as to what we do at Balbirnie Home Farms. If we go to the next slide, please, Christian. We farm here about 1,200 hectares altogether. You can see the numbers there. On the screen, there's a fair variation of crop types, quite a mixed rotation. Like most farms, we have the livestock element of the farm is very much remote, so to speak, from the arable enterprise. I don't think we're unique in that front. We're very blessed with sandy silt loam soils, so we're lucky in that front, and it gives us the option to grow such a wide variety of crops, and we have the opportunity for the high value crops as well. Next slide, please, Christian. So briefly, what we want to do, Fiona has given you an outline of what we're intending to do throughout this next 6 years of the strategic farm. But priorities here for the first year would be essentially finding out, what position are we in regarding our own soil health and the environmental landscape that we farm within. Are we good? Are we bad? Are we indifferent? Until we get these baseline numbers in there, we're not really going to know how good a job we're doing on that front. We might think we're doing a good job with lots of cover crops over the winter and not ploughing much of the ground, etc. But it's not for me to decide. I couldn't tell you whether it's finding benefits yet or not. Trial work, there's one trial going ahead this year in the first year, and that's essentially looking at nutrition. Can we grow a profitable crop without fungicides? Now, the aim behind this is to use bricks measurement to steer us as to whether there's something wrong with the crop. Is its health okay? Do we need to do some sap or tissue analysis to find out what we need to apply to keep this crop growing? Anecdotally, we've done some tramline work here on the farm in the last few years. We've had some great failures and some great results with it. But it would be nice to know that the theory that Joel will probably touch on later on is that we should, if you have a healthy crop nutritionally, which essentially comes from a healthy soil too, we shouldn't need fungicides, or certainly as much use of them to produce profitable crops. We really want to put that to the test and find out why can't we achieve it over a large scale, or maybe we can. Future ideas for the project. We do quite a lot of cover cropping here over the winter. We've started doing cover cropping in the summer too with mixed species. The idea is to try and integrate some livestock onto the land to build fertility and resilience into the system. So we need to look at what are the best species to grow. We seem very limited up here when growing cover crops in the autumn through the winter. There's not many things that can grow. We seem to have settled on a lot of rye, which fits having quite a lot of broadleaf crops in the spring, but it's a troublesome volunteer if you let it get through into the cereals, as we've seen over the last few years. So what choices do we do there? How much value can we get from the nutrient cycling that these cover crops can bring? Moisture retention on our sandy soils, these long dry spells. That's 3 years now we've had a long dry spell in the spring, and you can really see it hurting in the last couple of weeks before you get the rain. So we need to build on that and try and get ourselves a bit more resilient against these scenarios. And the big one, of course, we're all talking about now is carbon sequestration. Are we going to get paid for it? The government are driving us that way. How can we measure on that, and what can we learn? How can we sequester more carbon? Can we sequester more carbon? And I've already touched on integrating arable with livestock as well. So that's kind of a very brief overview of ideas that I have here for the strategic farm. We have our steering group who will pitch in other ideas, and we'll welcome to suggestions from the wider community. There'll be lots more ideas of things that we should be trying, could be trying, for the benefit of the whole industry, and I hope we can really make a success of this. So there you go. Thank you.
Perfect, thank you, David. Obviously, great to be working with you in a bit more depth here and trying to quantify the work you've been doing over the past few years, and again trying out new things to see where and when they do fit in the rotation. So that's great. So I think we're going next to Fiona Burnett, who is leading the trials with us, and I think we're going to start off with a poll as well. With that, Fiona. So we have the poll here. So which IPM measure do you rate the highest? And it should work. So it should be a case of just clicking the box. So you've got resistant varieties, longer rotations, more tailored pesticide use, or biological control options. A bit of a controversial one there at the bottom, but we've been discussing this every Wednesday with our SAC AHDB roadshows, so plenty in this to discuss. Certainly in resistant varieties seems to be very controversial at the minute with gene editing and everything else going along with it. So I'll give you another few seconds here just to vote on it. So this is going to obviously some of the things we try out over the course of the programme. It's great to get everybody's views on this, so we'll just close the poll now and see. Oh wow, there we go. Yeah, so quite an interesting one to lead on with there, Fiona. Resistant varieties obviously being people's key views on it going forward, which we did have a great chat on yesterday's webinar and Ask the Expert session. Plenty of work to continue with.
Yeah, no, that's given me a good starter, Chris, and not so far off what we might expect, that resistant varieties are a tool we're comfortable with using. The longer rotations is interesting because I think often we know that people actually really value the preventative measures like longer rotations, but maybe we don't get enough credit for that. So it's that idea that we don't take account of the negative decisions. You know, longer rotations, you've prevented a lot of problems. Right,
Absolutely.
I shall move into my presentation now and try and share my screen. So yes, SRUC are managing the home farm baselining and experimental work in this year one, which we're obviously, you know, delighted to be doing. And we're working closely with ADAS and NIAB, who manage the East and West strategic farms. So wherever possible, we've shared thinking and shared methodologies so that we can read across between the strategic farms. So what we're doing at Balbirnie is 4 linked work packages, and as David started to set out there, that idea that we start to link the health of the soil and the crop, the nutritional status, with the disease levels that develop or that don't develop. So you can see the 4 work packages there. So the first one, we're benchmarking the soil health. We're starting to baseline the pests and beneficials that are on the farm and in the fields. We're doing some crop health baselining. And then the one trial that we've got is this crop nutrition trial to explore the benefits of optimal nutrition and real-time nutrient testing. And as Chris has already said, this is where he likes to think you can beat the experts. So if I just run through, like I say, what I'm doing now is really just setting out what we were asked to do for the home farm, and you know, the different methods that we'll be deploying. So baselining the soil health, and this is to understand those 3 parameters, so the biological, the physical and the chemical status of the soil. So we'll be baselining 10 fields on the farm, using grids where appropriate, and we've used soil maps there to zone the fields and to pick out 2 or 3 zones, major zones per field, so that we don't you know, average over different soil types and so on. We'll be doing more detailed assessments for 5 of those fields and using the soil and plant health assessments. So across those zones, we're doing, you know, penetrometer readings, the visual assessment of soil structure and worm counts to build up that soil health package. And then we're doing very detailed assessments in one field, just to get a feel for how the soil health varies across you know, a region in a field. And we're doing that using the soil health scorecard, which is part of the SAC soil health test. And we can go on to talk about other, you know, parameters and ways of measuring soil health. But I think the key thing that we're doing here is just baselining this farm. And for all of us, it's that feeling, whatever you use, it's where you start and then how you start to track improvements. And then on the right-hand side there, you can see the list of assessments that we're doing. So obviously the basic nutrient testing, the routine testing you might do in a soil, so pK, pH and so on, the penetrometer resistance, the visual assessments. We're using earthworm counts within that, and again, I think Eric will come on to, you know, different, you know, ideas that you can measure nematodes and other you know, biologicals in the soil. We'll also be looking at some of the key things like moisture holding capacity, infiltration rates, bulk density to get a feel for any compaction issues, and we'll be doing the soil microbial biomass, potentially mineralizable nitrogen, total soil carbon and nitrogen. So a really good picture of the starting point for the fields on the strategic home farm. And that's just an illustration of the soil health scorecard, which I'm sure many of you will be familiar with, but it gives a range of, previous management regimes in the examples given there. And then the kind of traffic light system that says, right, okay, where you've used farmyard manure for 23 years, you can see that you've got good earthworm numbers, the visual structure looks good. There's an issue there with the phosphorus flagged up in red and so on. But that idea that, we can start to improve all those scores and track improvements going forward. The pests and natural enemies baselining is really interesting because, this is such a diverse area to get into. And again, what do you begin to count? And obviously, when you're thinking of pests and natural enemies and beneficial insects, you're looking at a huge range of functions there. So you've got, groups like earthworms and millipedes that are breaking down your organic matter. You've got pollinators, so your bumblebees, your hoverflies, and so on. And then you've got your predatory and parasitic, insects there, and the things like ground beetles and so on. So loads of different roles, different functions, and then that idea that you need to provide them with food and a diverse environment, not just at peaks in the season, but over the full season. So the way we're doing this on the strategic farm is to focus on 8 fields, using the centre of the fields and then using their adjacent field margins, because obviously they're very important in giving those diverse habitats and giving shelter over the the period of overwintering, for example. So a range of monitoring techniques, and we'll be trying to capture the beneficial plants and weeds, the pests and natural enemies, and those insect pollinators. And again, given a cast of thousands, we're going to focus on 2 key pests, so slugs, always topical for us in Scotland, and summer aphids. And then we're doing background counts of other pests that might be there, derived from pitfall traps. You see a little pitfall trap in the middle picture there. And we'll be doing samples from the canopy and so on as well. And then we'll also be doing direct monitoring from pre-baited stations, particularly for the slugs. And the next slide is maybe a bit detailed, but it just gives you an idea of the categories of pests that we're looking for. So looking for those ground-working beetles and spiders. We're looking at the solitary bee breeding success, and then we're looking at the pest control services that we're getting and seeing how well they're predated or otherwise. And then looking at the natural enemies and pollinators as we go on. And then finally, as I've said, looking at the floral and grass resources that will support those beneficials through the season. So you can see from the timings there that we're trying to work over the timeline of the season and deploying a range of assessments. Again, that's where we've started with the baselining. Inevitably, we've had to focus on a few key indicator species and a few key pests. So again, there's maybe discussion points around that. Getting on to the crop health baselining, what we're trying to do here is to baseline the crop health and then see how David's management practices on the farm affect that overall crop health. And very much with this, we're heading in the direction of examining that interplay between the soil health, the crop nutrition, and the plant health. And obviously, the strategy at Balbirnie is to reduce reliance on artificial inputs, which is very much in line with the direction we're being pulled in. And, we know we're losing plant protection products. We've got national action plans that we have to enact. So this idea that we may not go as low as no fungicides, but we can probably be more strategic and reduce those inputs if we understand better that interplay. So on the left there, sorry, the right, I've listed out the assessments that we're going to be doing. So again, we're focusing on very detailed assessments in 5 fields, but using the knowledge of the soil zones within those fields to look at 2 or 3 zones within those 5 fields to map out the plant health. So we'll be obviously tracking growth stages, the development of the crop. We'll be looking at biomass at a couple of key stages, and obviously within biomass, I'm lumping yield as our key biomass indicator at the end as well. We'll be looking at the nutrient status of the crops. So again, David has referred there to the idea that we do real-time SAP testing and compare that to, if you like, more conventional laboratory testing and really see whether we can be more reactive to the health status of the crop, because we do know that the minute, well, once it's evident that you've got deficiencies in the crop has already been toiling for some time and has already had the potential slightly capped. So the idea that we can monitor and react is a really interesting one. And then clearly
and I'll just come in there a minute as well, Fiona. You know, that is kind of the key mentality of the steering group with David as well, is that actually we want this live, we want it now, we want to react. We don't want to wait to yield and say, oh, well, you know, that didn't work. We want to know instantly what's going on in the plant. And that's what we're striving to do here, is to find out what's going wrong and if we can correct it with nutrition. And you know, fungicide as well. We're not for 1 minute saying it will only be nutrition. And if David's there, you know, you're welcome to come in as well here. We're doing a farm practice, and then we're doing our thoughts as a steering group as well. And these trials, yes, they'll ultimately be governed by yield, but they'll also be governed by the point at which they break down.
Mm-hmm.
I think would be my understanding, is that's what we want to know, is once it breaks down to disease, how much yield do we lose in, and what do we do from there on in?
Yeah.
There was oh, sorry, a bit of a delay there. A little interesting question on the soil side of things. In-depth analysis of the soil in terms of trace elements. We will have that from the soil results. You know, just people saying, are we going to look at in-depth trace elements within the soil? And I think the answer to that is yes.
Yes. No, absolutely. I don't know whether there was a particular interest in any one of those, but absolutely the idea that we move away from just the very simple, if you like, NPK, pH. And I mean, to be brutal, many people could do more of that and will be driven down the line of doing the very basic soil chemistry more often over more of our fields. But the idea that we start to pull in that, you know, enhanced detail and trace elements, you know, a very key piece. And again, a very key piece in what you'll be trying to track in your real-time testing as well, because it's often those trace elements that are the limiting factors and are now definite stress triggers for us.
Perfect, thank you.
No, it is a really interesting idea. And again, for reasons of practicality, you know, a lot of our programs, be it on the nutrition side or on the fungicides, are very fixed on key timings because that's practical for us. We can forget about it in between times. And it will take more management brain to be working in real time like this. But absolutely the scope that we can better target what we're doing and really tailor it to, you know, even if we could tailor it on field levels, but what we're doing on the farm here, you know, whether we could tailor it to the different zones within the field is, you know, a really interesting piece of work to be working
Absolutely. On. I think there's much more passion within the industry to know, you know, we've got chemical names there, but actually when you start and look at the chemical formulation behind the name, it's only then that it starts to get really interesting, saying, well, what are you actually applying and how is this manipulating the plant? You know, and there are, you know, other effects of fungicides as well, aren't there, on the soil? So it's just trying to take all this into account.
Yeah, no, and again, that interplay that you know that, you know, clearly the health status of the crop, if it's deficient, it will be, you know, stressed and perhaps more vulnerable to some diseases. But otherwise, you know, we've never tried to look at the whole piece and the link between, you know, the microbes in the soil and, you know, what it triggers in terms of host responses in the plant. That complexity is huge. But to start unpicking it as we are is, you know, again, a really interesting thing to be working on, Chris.
Definitely, and especially having it ground traced in Scotland as well. We're very happy to have it up here. So
yeah, no, I'm sure we'll find a huge range of answers just within fields, never mind on this one farm, never mind when we start to roll it out. But you know, it's you know, exciting to be doing this type of work.
perfect.
I'll maybe get back to the slides now. Yes, we'll be monitoring the disease again as we go through the season, obviously reacting to what appears, doing many of what you would judge to be the standard assessments, so looking at both the foliar and the stem-based diseases. And as Chris has said there, you know, noting the times when the crop starts to break down to disease, whether that's different across the zones and across the fields. And then the final thing I wanted to touch on is the idea that we will score the management practices on the farm and within the fields and give it an IPM score. And when we wrote the proposal for AHDB and suggested this, this was a research tool that wasn't available to growers, but just last week we launched the Ipm Metric for Scotland. So this is now available on the Plant Health Centre website and is a tool that everybody can use. Many of you will have filled in the old style IPM plan as part of your quality assurance schemes. So this is a refresh plan which lets you look at your management practices. It's just a tool for you. Nobody else is looking at the data, but it lets you see how your farm scores. And we'll be doing this for David on the Balbirnie farm. And we would anticipate that that farm will score quite highly on the scale. But kind of where I started my presentation, this idea that we're not doing nothing at the moment, actually many of you are using quite strong rotations and measures that actually give us quite good IPM starting scores. So you see there that the mean score in the kind of prototype farmers that filled in the survey was about 67%. English and Scottish growers quite nicely scored higher than the Irish farmers that were involved in the survey. But you can see there that we have some quite high scoring farms. So it's really interesting to start to unpick what those farms do and then what perhaps you know, other farmers could do to improve their scores. But very much again, in line with what we're doing at the strategic farm is that idea of baselining, knowing where we're at, knowing what works well, and then improving the situation. And some of the things that we know those high scoring farms are doing is that very clear link. The more they understand or know about IPM, the more they actually implement it. And the idea that the knowledge exchange and information sources are very important. So very commonly, and a little plug for the strategic farms here, the people that score highly when it comes to IPM are people that go to discussion groups, go to open days, come to trial events. So that's, you know, maybe a message to kind of finish on that the idea of engaging with things like strategic farms is a key way of improving things all round. So I'm not sure if there's a few more questions come in there while I've been talking, Chris, but you can let me know.
Yeah, there's certainly more than one or two come in. So as I say, we will try and get through them as best we can, and we will strive to answer all of them and put on the website. So a couple of them there, just to clarify, pH, how do you soil test which system? SAC, so is it Morgan's or Oilseeds?
Mm-hmm.
And I think we're very much going down Morgan's at SAC, so that we are comparable with the rest of general farms, if you like, in Scotland, to try and keep it as relatable as possible. The other question that we did have there, are you also integrating any farm scale habitat baselining to assess quality of habitat for beneficial organisms and make sure to maximise overwintering and potential early pest regulation at the start of the season?
Yes.
Very interesting question. Absolutely. And David, there's actually other parts of the studies been going on at Balbirnie as well in terms of the wider environment. So we've had quite a bit going on there, which Johnny and David are undertaking as well. So I think we'll just move on just now so that we can get stuck into Eric Anderson's presentation as well. There'll be a lot more questions from that, no doubt, as well. So we've got a poll just to kick us off before Eric starts. So, wait a minute, my question box broken it. So what is a grower's primary objective when growing a cover crop? Now this is very topical here, obviously, just now. So biomass production and organic matter improvement, pest or pathogen control, improving cultivation and soil structure, preventing soil erosion and pollution, increase biodiversity and create wildlife habitat. So this should be very interesting to see from what perspective we look at it. I think from my side of it, you'd really want all of the above and a nutrition store. What wonder crop or cover crop could be. I think, as we look at things, probably we are heading more and more in this direction. The AECS scheme has certainly been interesting to watch and eye-opening to those that are in it, to see the overwinter stubble plus the green manure crop, just the difference you get after that. So if we just have another wee second there and close the poll, Christian, and we'll see where we come back. Okay, fairly even spread there, Eric. Not so much on the pest or pathogen control, but yeah, interesting. I didn't think there'd be as many people looking at it for cultivations and soil structure, but I suppose it's becoming the norm in the potato industry to try and have a bit more of a cover crop in there pre-potatoes, which is good to see.
Okay, thank you, Chris, for doing that warm-up for me. You should be seeing my screen now.
Yep, I think again there's a little bit of a delay, but we can see your slides, so yeah, you can carry it.
Okay, thank you. Well, when I've conducted that poll previously, we do see the consistent top choice is improving cultivation and soil structure. It's interesting to reflect that little cognizance is being given to the other ecological benefits potentially associated with cover crops. So we just need to perhaps widen our horizons and widen our minds. Looking at what's on the horizon at a political context, evidence is certainly building of a new agricultural Scottish policy from several of these documents you can see here. Carbon auditing. The UK will host the United Nations Climate Change COP26 conference in Glasgow in November 2021 this year, and the UK will aim to cut its carbon emissions by at least 68% of where they were in 1990 by the end of 2030. And the government will use this opportunity to build momentum for repurposing of agricultural subsidies to protect and enhance our biodiversity and build resilience against climate change across agriculture, land use and food systems. And domestically, reaching our net zero target is one of the government's top priorities. Additionally, we know that Fiona and our team at SRUC have been developing the carbon footprint tool, AgriCalc, that's been developed in conjunction with RESAS and Innovate UK Technology Strategy Board. And this tool will initially be used through the monitor farms and through the AHDB strategic cereal and potato farms to help build a better impression of greenhouse gas emissions from businesses and identify where there are areas for improvement. Additionally, within these documents, there is some common threads running through. We can see water quality, biodiversity enhancement, sector collaboration through data sharing, and soil analysis requirement. Now, there's a lot of misunderstandings and misconceptions at present, I think, within the farming community and the political community. Facts have been grossly oversimplified, and we collectively need to inform where government policy choices should and can be driven. Public debate is frequently dominated just now by silos of self-interest. Now, farming is frequently vilified and is a soft target for the media. Remember that farmers are a resource and an ally in climate change challenges. There are opportunities for transformational changes to reinvent ourselves, to improve ourselves as an industry, to adapt technology and to adapt land management practices. As agriculture as a whole, we need to take responsibility to share data in a transparent and holistic way and to demonstrate in a constructive manner where and why we see improvements being made. And that's all part and parcel of the strategic farm. Farming needs to embrace the science and measure things properly. Knowing about the problem is the first step in actually tackling it. Now, coming back to carbon dioxide, it's got a half-life of 27 years, which is the equivalent of a residency time in the atmosphere of 39 years. But the big problem comes in the transfer of carbon dioxide from the atmosphere and dissolution of carbon dioxide in the deep ocean, which effectively means it has got a global warming potential of up to 500 years. 500 years. And using a bathtub analogy, with carbon dioxide, we need to get to net zero before we derive the benefit and stop the bath from overflowing. In the grand scheme of things, there is often an oversimplistic or oversimplification because nitrogen and carbon cycles are strongly interlinked. There's a positive correlation between soil organic carbon and nitrous dioxide fluxes arising from the biocoupled geochemical cycles of carbon and nitrogen. Now, whilst carbon sequestration is easy for all to understand, as you increase soil organic matter, so biological activity increases and nitrous oxide gas release is also increased. Thus, other greenhouse gases are arguably more important than CO2, especially in the context of agriculture. Nitrous oxide is a powerful greenhouse gas with a global warming potential that is 298 times that of carbon dioxide. It's emitted from nitrogen-based fertilizer and animal wastes added to the soil. And once created, nitrous oxide stays in the atmosphere for 114 years. Now, nitrogen derives productivity in agricultural systems, and yet it represents one of the biggest challenges to sustainability at a planetary scale. The Scottish Government have brought together the Arable Climate Change Group, which will report in early March this year. The Scottish Government on the 1st of December in 2020 released a consultation on proposals for establishing a Scottish nitrogen balance sheet as a method for determining a baseline figure for nitrogen use efficiency in Scotland. We currently live in a world where yield is derived from external inputs. Modern arable systems are good at producing food with agrochemicals and artificial fertilizers, but can we transition to reducing inputs in a sustainable way without reducing farm incomes? And that's important. We've got to maintain farm incomes. The challenge will be balancing crop nutrient requirements whilst minimizing losses to maintain a sustainable environment and economic benefits. And the starting point for achieving all this is high nitrogen uptake efficiency and creating an environment that enables the roots of the growing plant to proliferate through the soil mass. Leguminous crops have high nitrogen use efficiency because they gather and store nitrogen in their bodies rather than releasing it into the atmosphere. And amongst cereals, oats have a high nutrient use efficiency, and certainly higher than that of wheat or barley. In the near future, therefore, carbon footprinting will be driven by nitrogen use efficiency metrics. The amount of carbon exchange between the carbon pools of air, land, and living things, rather than soil organic carbon content, will be the critical factor in soil systems. Now, the response of nitrous oxide emissions or emission factors is related to both the quantity of nitrogen that is applied to a field or to the soil and to rainfall in a geographical area. The higher the rainfall, then the higher the emissions of nitrous oxide. And we can see that at a UK level, we've got spatial disaggregation of nitrous oxide release. The drier East Coast areas will release less nitrous oxide than the West Coast areas because of rainfall. And the rainfall is related to the pore space in the soil, and we know that where we've got pore space greater than 60% saturation, then it will increase nitrous oxide release greatly from the soil. On occasions, you'll see nutrient use equations where nitrogen output is simply compared with applied nitrogen as an input. This can be satisfactory, but it assumes that within a crop rotation, soil available nitrogen is at a constant, and you'll see more of that on my next slide. There's much greater complexity there in reality. Evaluation of crop performance and nutrient use efficiency needs to be considered. So that is G in this equation. We've got the genetic variation and improvement in varieties over time, varieties. We've got E for environment or environmental conditions such as climate, including annual variability and location, together with the agronomic and management practices. And it's this combination of these parameters and the interactions of G by E by M, which will determine the sustainability and secure crop yields, as well as nutrient use efficiency. And I believe that the strategic farm here has a great opportunity and for stakeholder engagement to evaluate these measures that will make greenhouse gas emissions targets achievable, but will also enhance the arable sector's reputations as one of the most efficient and productive in the world. We are very, very fortunate in the UK in having the likes of the Broadbach experiment at Rothamsted. That highlights many factors influencing both yield and nitrogen utilisation in wheat over the last 175 years. Practically, management and yearly variation over all of that 175 years. Now, interesting, if you look at the left-hand side graph there, you see the long-term versus short-term nitrogen response and how that differs from the UK average short-term response and long-term nitrogen response from the Broadbach data. Modern fertilizer trials assume a constant state of mineralized soil or mineralized N from soil. Look at the difference in yield at N0 rate in that left-hand side graph. And that's important if we're changing the dynamics actually on farm. Now, farming activities account for about a quarter of all greenhouse gas emissions in the production of either wheat, in this instance here on this graph, or indeed with potatoes, with between 25 and 31% of those being produced from the production of fertilizer. And whilst agriculture is responsible for 11% of UK's greenhouse gas emissions, it's only through the industry whose primary function is to remove carbon dioxide from the atmosphere. That's farming. We're removing carbon dioxide from the atmosphere through photosynthesis and the production of energy-rich products. Agriculture there has a great opportunity not only to reach net zero emissions, but uniquely, it has a potential to go net negative. That's a big win, and we should be flag waving on that. Typically, a wheat crop of say 10 tons per hectare of grain produces 18 tonne per hectare of biomass above ground and an estimated 1.8 tonnes in roots below ground. Around 46% of this biomass is carbon, and carbon makes up 27% of the carbon dioxide. A typical grain crop therefore fixes around 34 tonnes of CO2 per hectare. However, this carbon is released back into the atmosphere again when the grain is consumed and when its residues decompose in the soil. And unless there's some of this carbon is retained, for example, with stable soil organic matter, then there's no net reduction in the atmospheric carbon dioxide. So carbon fixation by crops is not normally considered when we're looking at greenhouse gas calculations. Interestingly, from a crop perspective, what matters is the greenhouse gas intensity per tonne of grain. So the emissions per hectare are divided into the grain yield. And this graph clearly shows the dominance of nitrogen fertilizer on nitrous oxide emissions and the carbon intensity of cereal grains. Together, these account for far more than 70% of the total. And there's some variation between farms in the costs from the non-nitrogen fertilizer and from machinery use, reflecting difference in the establishment methods. And we can get into all sorts of debate regarding inversion versus non-inversion tillage. When we look at the soil, and traditionally we've looked at soil analysis as a guide for the nutrition of crops, the actual practical usefulness of laboratory values as a basis for judging phosphate and potash fertilizer dressings, however, can only be judged from their relationship to crop performance in the field. Now, as far back as 1969, Reith, in a published paper, stated that the soil analysis regularly accounted for less than 30% of the variability in responses to crop yield. And that's important. Far too much emphasis is weighed just now on soil analysis data. The crop may not be able to absorb the phosphate that you've measured as a metric in the soil analysis. Coming now and reflecting on ecosystem services, ecosystem services deliver multiple benefits, and many of those are associated with farming. They can be defined as the ability of the soil to perform ecological services or functions essential to people and the environment. Fundamentally, resilient soils can adapt more readily to variation year on year and to climate change, to high rainfall events. And if we get our soil husbandry right, we bring together the conditions that will create the opportunity to deliver yield. How should we actually measure soil quality? And there's a lot of debate at present. Fiona has shown some metrics that she is proposing to use to measure soil quality. I think we've got to bear in mind that nematodes are the most abundant metazoan on Earth and occur in every habitat. Soil bio project that I was involved with about 2 years ago looked at testing and calibrating using nematodes as trophic groups, as a matrix, or as a proxy to measure and quantify soil quality across a range of common UK agricultural soils and crops. And the benefit we found from that project of using nematodes as indicators of soil health were their abundance. There are millions of those in every square metre of soil. There's high richness or high species richness of nematodes. They're tolerant to their tolerance to sensitivity ranges are quite large. They've got low mobility, so they don't tend to move around the field very far. Their cycle times range from a few months to a few years. And importantly, they have an interstitial mode of life. So they do reflect what's happening between soil particles. If we consider that and contrast that against earthworms, there are 27 species of earthworms in the UK as a whole, and 70 to 85% of those earthworms that we find in fields are juveniles that cannot readily be identified to a species level. We know that in Scotland, we've got 4 cryptic lineages or species that may be distinct ecotypes or biotypes, and hence can confound research based on their morphotypes. Basically, those 4 cryptic lineages, you can't visually tell them apart. They have plastic trophic group status that doesn't take account of the earthworm function. So you cannot separate them into the epigeic, those in the surface, the endogeic, those in the mid-soil levels, and the anisic earthworms deeper down the soil profile.
Eric, just coming in there, this is fantastic research that's in there. Now, when we're ground truthing this, now, you know, we've known we've got the visual health scorecard there, which is a fantastic way of going around your farm and starting to understand things. Now, this is quite in depth, but where's the correlation here with actually, you know, again, my very simple mind wants to influence this. So, you know, we've had a soil test that's very simply N, pH, P and K, and we can influence that by very simply the amount you put on. Funnily enough, 5 years later, you pick up in the soil test. But where are we now with this? Because presumably the cover crop actually is the biggest influence, you know, roots in the soil is what influences that, is it?
Yeah. From the studies we undertook within the Soil Bio project, we undertook 6,000 soil analysis across the whole of the UK. And what we found was that the cropping, the management of that crop in terms of the mechanization and the cropping in the previous 2 years were having the greatest associations or influence in the soil quality as measured through the different trophic groups of nematodes. And it was very, very repeatable. So there's lots of things that we have historically measured by chemistry. We haven't been so acute at measuring the soil biology and how those changes transform or take place over time. We know, Chris, that making the wrong choice in cover crop can increase the population density of plant parasitic nematodes. So on this slide here, we see Pratylenchus nematodes in particular can build up rapidly when green manures are applied. And unless one is using proven cover crops with biofumigant properties, then you can unintended build up the population of Pratylenchus and parasitic free-living nematodes. We know that there's well-documented cases of disease and complex disease interactions between Pratylenchus and verticillium wilt in potatoes, causing potato early dying and significant reduction in tuber size and yield. And you know, we have got potatoes in rotation here on this strategic farm. So increasing fresh organic matter with green manures can increase soil-borne rhizoctonia as well. So this emphasizes the importance of thinking through what we know and what can be achieved and what we're trying to achieve. If you're looking to improve soil structure, the additions of amendments such as FYM or cover crops are good, but you have to consider at a much deeper level the unintended consequences.
Eric, can you assume that what eats the nematodes? Something must live on the nematodes. What's the kind of apex predator? Is there something further up the food chain that as a producer and land managers we can look at with a spade that tells us, well, there's lots of beetles or worms or what have you, and they're supported by a network of huge numbers of nematodes, which feeds back to your research, which allows us to dictate at a very basic level a degree of our soil health. Is there any correlation or work being done with that at all?
Well, you see from one of my earlier slides, then at all levels of the soil biology, then nematodes are involved. You've got 5 trophic groups of nematodes. You get nematodes which feed on other nematodes. You get plant parasitic nematodes, but they only account for about 15% of the total nematode population. You get nematodes which feed on plant bacteria. So you've got these different, and you've got omnivores which feed on everything. So you get these different groups of nematodes, but the only way to reduce nematode populations in the soil is to use the likes of cover crops with either biofumigation or partial biofumigation properties, particularly those with 2-phenyl ethyl isothiocyanates, which is basically mustard gas, which will reduce the population levels for a period of 2 to 3 years. But there
So
are.
Eric, if you go on there, so if we're saying like unintended consequences of cover crops applied to more single species cover crops as opposed to multi-species stanza cover crops, so like in essence, are like the more species of crop in the ground, the better. Is that where we're getting to?
Not necessarily, Chris. The more species you put in there, you may just cause confusion, and you don't know which one is doing the right one or the right function. So putting in an appropriate mix, yes, I wholeheartedly would embrace that philosophy, but there needs to be a lot more work done on what that appropriate mix is, and what are we trying to do and achieve in that mix. There is a lot of guidance out there just now that puts in a scattergun approach, and you get very complex 10 species mix cover crops. That's just too much. Know what you're trying to do, achieve, and choose something that's appropriate.
Completely agree, and I'm thinking, I'm just dulling it down a little bit here to my level, but we think of the soil as a store, and that store, is measured by just now organic matter content in my head. New Zealand's at 10%, we're at 1, 2, 3%. To try and increase that organic matter store is what we're being led to believe, and that is the way that also anecdotally would hopefully produce more nutrient-dense foods without the need of fungicide if we can get to this holy grail of organic matter. How you do that versus mineralising enough nitrogen to grow a crop to take it through to the point where it can actually access the store of organic matter, to me, is the trick. But we need to understand what we're putting into the store. Would that be right?
Yes, and also you will not, if you look back at the Rothamsted and Broadbach experiment running for 175 years, the different rotations that have been run there and different organic amendments, you find that there are changes, subtle changes that take place in soil organic carbon and measurements of soil organic matter, typically measured by loss and ignition. But that takes place over a very long time scale, over a 10-year plus period, that will not be measured within a rotation or between crops. You also find that the additions of organic matter, you raise levels to a new plateau, but it will plateau. It doesn't continue to increase on a linear scale or a linear fashion. You increase soil organic matter, but it's the functionality of that soil organic matter that is important, not the organic matter per se. Not all organic matter is equally valuable. You got to think about the cycling of that organic matter within the soil and how many bugs and beasties it is able to feed. You've got a large quantity of biota underneath the soil, equivalent to 30 to 50 sheep in terms of biomass are munching away underneath the soil. You've got to feed those 50 sheep underneath the soil in a metaphoric sense, otherwise they will die.
Yeah, absolutely, and we've seen that, anecdotally, and the 3-crop rule, for all it's a disaster to some, it's been beneficial in terms of soil health. That continuous spring barley is horrific for the soil, isn't it? Potentially with artificial inputs. And what we're trying to get back to is this: what's in the roots, and how can we then take that and influence the disease portfolio? Because we've had this great time that my lifetime's only been at the end of it, where you throw nitrogen on, you get a massive crop, but we're not going to be allowed to do that anymore.
No.
So really, how are we going to grow these crops? And applying organic matter is not the same as growing it. And one of the things, I listened to snippets of webinars, but I think that I heard the figure batted about that when the usual old adage of chop the straw, it's better for the soil. It's 7 years before you see the benefit of that.
Yeah. We're working here on long time scales, and there are actions that arable farmers can take to improve soil health. We can undertake an inventory, perhaps, of natural capital in the farm by knowing what you have, quantifying and undertaking evaluation of your assets on the farm, and perhaps starting to have a wider public engagement. There are 3 actions that I think we can take at an arable farm level that's going to improve soil health, connect field margins and hedges, and protect and enhance permanent habitats. You're going to be paid for doing this in the future. So, at a farm level, area and condition of semi-natural habitats and environmental assets is going to be important. Undertaking an environmental audit, and Fiona's already talked about that. And perhaps piloting an outcome-based approach. I know Nature Scotland are leading on a project on that at present, looking at the vegetation, the pollinators and bird surveys. You mentioned carbon auditing, but diffuse pollution in the 4-point plan, soil test and VES or visual soil assessments are going to be important, but let's not forget about the natural capital. In the middle of this slide here, we see a piece of work we did on the strategic seed potato farm in Scotland this year, looking at cover crops and running wildflower mixtures through the tram lines, on every tram line through the potato field. And you can see that there was a huge difference in the number of invertebrates per sample in the two habitats, with a significant difference of more than threefold in the habitat that was associated with the cover crop rather than the potato tramline with potatoes growing in it. So we can certainly significantly influence the habitat and the biodiversity of what's happening on our arable farms. But why, as arable farmers, is biodiversity important? It's all the pieces of the jigsaw are present in front of us, but until someone puts all these pieces together, it's difficult for most people to see the whole picture. Healthy soils support biodiversity, food and shelter for farmland birds throughout the year. We've got pollen and nectar for pollinators in the spring and in the summer, and we're creating habitats for invertebrates, mammals, amphibians, arable plants, shrubs and trees. And in a biological system, these impacts are generated over time, as you've pointed out, Chris, through dynamic and complex processes. And crucially, these steps are required to ensure that the change is successful. But the first step is the important one on the journey. All this seems impossible until it's done. And where the soil is deteriorating or inputs are increasing, can we use soil armor by keeping the soil covered throughout the year, together with a wider and more diverse rotation, encouraging beneficial soil microbes? There's lots
just
of—
a little question, Eric, in here at the same time. So as soil processes, there it goes, a lot of questions. As soil processes take time to change, what soil parameters are you choosing to measure change? Organic matter and food web can take 3 to 5 years to catch up, as we're discussing. How do we measure management impact over scales that are helpful for farmers but also realistic?
Well, you will not be able to manage to measure these dynamic changes at a biological level, unlikely within a cropping season. It will be 3, 4, 5 year cycles before you see the dynamics changing within the soils.
Yeah. So the difficulty then comes, thinking about an outcome-based scheme. How do you reward farmers for beneficial soil management when we might not be able to measure change quickly? So how are we going to measure a healthy soil, and then how are we going to put in the metrics of change to influence that?
The metrics of change will only be over longer timescales of 5 and 10 years time, but you've got to have a baseline that is important. And politicians just now have grasped the opportunity to look at carbon. Soil organic carbon, loss in ignition is relatively cheap and easy to measure. It's a simple metric, but we must be cognizant of the fact that it is a simple metric, and what's happening in the soil is much more complex than that. And perhaps Joel will reflect on that within his presentation. There's lots of abatement methods and mitigation that we could include, and I've listed some of those there that we can look at an arable context, but these are not going to make changes, as you pointed out, Chris, in periods of 6 to 12 months. We can encourage crops and varieties with improved nutrient use efficiency, so you have varietal improvement there, and that includes disease parameters as well, and disease resistance. We can use manure and fertilizer management technology to place fertilizer appropriately. We can use nutrient management plans for all land, not just those within NVZs, and use a recognized fertilizer recommendation system such as RB209 or the SRUC fertilizer management note.
Yeah. Fiona's just coming in here, Eric, at this point as well.
Yeah, no, I just wondered we could come back to it, Chris, but the difference between the kind of national inventory numbers, which government and policy will be looking at, so kind of how we perform as an industry versus the on-farm calculations, which can be, there are plenty of win-wins and things that cross over, but there is a distinction between the efficiencies you'll make on farm and that wider national inventory. So we could maybe come back to that.
Yeah. There's lots of things.
Key question there, but how are we going to measure a healthy soil?
It is fundamental, and we've been debating this for the last 20 years. As what does healthy soil mean? Just measuring chemistry doesn't encapsulate it. You got to look at the physics, which is expensive. If you look at soil water release curves, for example, those can take up to a year to actually run in the lab. Measuring penetrometer resistance is only as good as where you're measuring it in the field. You need to take soil and soil cores in little plates back to the lab to do that effectively at different controlled soil moisture levels, and it's only through doing that that you'll get accurate reflection of the results and the information. So it's
which is great. Now, the simple world, according to Chris, and my philosophy is, final in my head. But what, if we looked at the preceding crop, which I know Soil Essentials have been doing with their potential historic index, the HPI, historic potential index, get it right? Is that a better way? Look at the crops and actually assess. So, are we using a cover crop as a signal to say, what's in the leaf or what's missing in the leaf? Does that feed back down into the soil and say, well, we're short on, molybdenum in here? Is that the limiting factor? We need to actually start and look at applications. Is pH, when we apply lime, can the soil actually use the lime, or does it just flush straight through?
Okay, lots of questions there, Chris. How much time are you giving me? The HPI, historical potential indices, it's a surrogate for biomass, really, spatially across a field. We have an advantage in that there are satellites moving across every field in the GB every other day. And therefore, there's lots of data sets there are available for this year and for historical context. So we can very rapidly build up a picture of how biomass is varying spatially across a field over a number of years and a number of crops. And that gives an insight as to where you should be looking within the field. Is it a problem that can be managed, and can you mitigate, and can you undo some of the constraints that are there? Or is it something that is unmanageable and you've got to accept, and we can put that into a diversity habitat that's not going to be cropped? What we did find from the Soil Bio project was that using HPI and biomass across a field was that the nematodes were reflecting the soil physical properties as well as soil chemistry properties, and it was much cheaper and quicker to turn that around, because we can take a soil analysis for nematodes, run that through the lab using TLFRP in order to identify the nematode species and get an answer within 10 to 14 days. So that is the way that science is heading us, that we can actually tie many of those metrics together and measure soil quality as a collective of physics, chemistry, and the biology within the soil. It also reflects the soil properties and physical properties of the soil, and I must emphasize the last point on that slide there is avoiding soil compaction. Loosening compacted layers of soil in the topsoil and the use of cover crops as deep tillage for deep compaction, because with the axle weight we're using on vehicular traffic and tractors nowadays, we're causing deep compaction through the axle weight of the tractors, irrespective of what boots you've got on in terms of tires, well down the soil profile that is beyond the cultivatable depth. And we found that in 50% of farms and fields that we measured in Scotland, there were constraints to crop yield coming from soil compaction.
We'll not mention potatoes in this then, shall we?
We can debate that if you wish, Chris, but we can also debate whether we should be encouraging more legumes in the rotation to recycle nitrogen and to increase nutrient use efficiency. There's much debate, however, at present whether there's a net loss of nitrogen is less where legumes rather than other crops, and whether some of that is lost during the decomposition of both legumes and with other crops. With clever management, cover cropping works at a number of different levels, and if you plant the right species and variety, you can reduce the spread of disease in a crop, you can suppress free-living nematodes, and you can create more resilient soil architecture. In this context here, utilizing a flying focus sheep to graze off a cover crop can further increase organic matter and return nutrients back to the soil again. And I'll just finish on two quickly last slides here. There's increasing interest in stimulating saprotrophs to suppress plant pathogens. This is without using chemistry, using natural saprophytic fungi and their antifungal bacteria to suppress plant pathogens. Basically, by increasing soil organic matter, can we increase both the microbial biomass and microbial activity in order to generate suppression or at least buffer diseases within arable crops. I've probably given you more than enough information there just now to fry most people's brains, but I think it should open a whole debate, Chris.
Yeah, absolutely, Eric. It's created a lot of questions, and trying to ground truth it as well. I mean, one of the the bits that a lot of us have been doing is this intercropping and understudies and things like that. Where do you see intercropping coming into play in the future?
The problem in Scotland, the problem in a Scottish context is that we've got a very narrow time frame in order to establish crops, and we have challenging soil conditions in the autumn and early spring period. There's no doubt that intercropping, we can achieve it. We are successfully establishing the likes of clover into wheat crops, and we have tried to retain the clover through the stubble before sowing beans into that same stubble. And you'll notice that clover is quite challenging to kill off, even with glyphosate. And we can take that understory of clover through to a bean crop. So yeah, we are more than experimenting. We're using that in practice on some farms that are leading lights in this sort of practice.
Yeah, absolutely. And it's very difficult, as you point out. We're in a high intensity or a high intensive rotation, if you like, of cropping and cultivation and applications of nutrition. I mean, how do you back off this? Where do you go? Because some of these crops, bring in, the linseed, for instance. I mean, it can be rack and ruin, or it can be riches, but there's no in between with that.
It requires a total change in attitude of the grower and of the agronomist, and that's got to be reflected as well in the economics of the outturn. At the end of the day, it's still got to be profitable, but if you're not spending as much money on agrochemicals and or fertilizer, you don't need as much yield in order to still be profitable. So it looks different. It may not be as appealing to the eye as a completely homogeneous crop of wheat or spring barley, but yeah, if you change your mindset, then it can be as profitable.
Yeah, absolutely. And I suppose from our side of it, in terms of our baselining, it'll actually be repeated in year 3 and year 6. So we're going to really be constantly measuring, all the metrics in the soil there. And we'll be running other soil assessments alongside the base of what SAC is doing with Fiona. And Eric joins us on the steering group with Donald as well, to have a good thrash at this and to try and challenge it. So, I mean, obviously we're going to get into the glyphosate debate here and the effects of glyphosate on the soil, but surely that's a game changer as well with all of this for all farming.
If there is some point in the future where glyphosate is banned, either pre-harvest or in stubbles, then that will have an adverse effect on our choice of cultivation practice.
Yeah, absolutely.
If you're using non-inversion tillage without the use of glyphosate, it becomes extremely challenging.
Yeah, without a doubt. And how do you think that will affect the loss of, you know, the cropped area as well, in terms of, you know, if we do lose decoupled payments from production subsidies, if you like, you know, do you think we'll actually see, you know, this break in the rotation that we need or we're asking for?
We know that in England they are moving faster along this journey than we are. They are losing. If a farm has between 50 and 150,000 pounds of BPS payments, they will lose 65% of those payments in terms of value over the next 4 years on a sliding scale basis. Scotland is not entering that transition period until 2024, but we will ultimately have to get to the same point in time by 2028. So without piloting some of these measures and practices and finding out where under Scottish climatic conditions they will work, then we are actually at a disadvantage to the English scenario just now. In England, they've got pilot studies looking at environmental land management schemes in order to see whether taking different management practices are beneficial and financially achievable on farm. There is no measures in place just now to pilot those same sort of studies in Scotland, but I think we're missing a trick. If we wait until 2024, we're starting behind the curve and we're starting too late when we've got different climatic conditions, different soils, and a wholly different set of challenges with a constrained, you know, season in Scotland.
I would absolutely agree, Eric. I mean, this 2024 or 2022, whichever one you want to look at, it's a brick wall. You know, now historically, when we've been thrown that by Parliament, you can go round the wall. Is generally, you know, that's what I tend to look at in terms of subsidies. This one, I don't see it. You know, personally, I think it's a brick wall. And there are a few, and that's the aim of the strategic farm, is to get this slope up to the brick wall so we can maybe just hop over it. And that's where we need to know how to measure what we're doing. But, you know, to continue on the same path.
I liken it really to a an obstacle race. Yes, you gave the analogy there. You can go around the side of the wall. What we actually need to do is collectively work together as all partners in the industry, so it's a team approach, and we can actually get over that wall. There is no going around anymore. I agree. We've got to go over the wall, and that's going to require a much deeper understanding of the biology, of ecology, in terms of agronomy and agronomists, and indeed farmers.
Yeah, absolutely. No, well, look, thanks, Eric. We've given you a bit of grilling there, you know, and you'll be here at the end of the session if we do get time after Joel as well. So, you know, thanks very much for that to everyone else as well. Anyone who has joined us late, look, if you want to stick your basis and fax numbers and roster numbers in the chat function, we'll pick them up and get you some points as well. And we're going to bring in Joel Williams, who's joining us from across the ocean today to try and give his little bit on it as well, which might turn into a big bit. So we'll hand over to Joel now. Oh yeah, we've got the poll first. Well done. So Paul, when growing a crop, do you use a protecting strategy, a reactive strategy, nutrition-based strategy, or you're not involved in making that decision? And I suppose behind the scenes here, you know, ultimately we see the French decision to politically decouple advice from selling products because it's believed to be a conflict of interest. So I suppose that's already out there. You know, the French are quite well ahead in terms of glyphosate being banned, other than a regen ag scenario as well. It's lots of controversial stuff there, without a doubt. So, yeah, a bit of a question on agronomy here. So if we close the poll down now, Christian, let's see what we get. So very much still a protecting strategy. So that's definitely been what's been forecast to us in the past. You know, in terms of all the meetings we go to, is that, you know, we don't have the chemistry to react to it. And I suppose what we're suggesting here is we want to inform ourselves a little more intensively so that we can try and react to it. And if we have to reach for the can, you have to reach for the can. There's no problem with that at all. It's still there anyway. But very interesting poll. Okay, Joel, that's one to hand over to yourself there to take that on.
Thanks very much, Chris. Apologies, my webcam doesn't want to behave this morning, so perhaps that's a good thing in the end. It's quite early for me here. So, okay, well, I thought I would pick up, I don't have too many slides, so maybe we can whiz through this and open up for some discussion. But I thought I would really pick up on this point about nutrition-based strategies and what role they might play and just some of the principles behind that. And then I'll wrap up with just also a few thoughts on some of that transition kind of discussion. So let's go here. So really it comes back to this slide for me outlining photosynthesis. And the point is to say that, of course, this is how plants grow. This is how they produce their biomass. And you know, as we all know, they breathe in that carbon dioxide and take up water. And then they use the minerals, the nutrients from the soil, the essential macro and micro minerals as part of enzyme systems, which help to catalyze, well, the very first product of photosynthesis, which is glucose, that sugar molecule there. And of course, they release oxygen. And the point is to say that they can't perform that step without those mineral catalysts. And equally then what the plant will do with that sugar molecule, that building block, it will then equally begin to stitch those together and form more complex sugars and more complex carbohydrates. And then equally a whole range of other plant compounds also stem from that very first building block. So, okay, they'll link in some nitrogen, some sulfur, build amino acids, build proteins, fats, lipids, waxes, and then a whole host of other compounds, hormones, growth regulators that they produce themselves. And then moving into some of the immune-related compounds, so protective compounds, defense chemicals, various volatile organic compounds that can deter or attract in beneficials and pests. Equally, they can build physical barriers, calluses on their skin, harden up their skins, and all of these things ultimately help to kind of induce a systemic resistance within the plant. And so it's really just to say that, once again, that second transition there, the second stage at the bottom, again, it requires the macro and micro minerals acting as part of these enzyme systems to catalyze the production of these secondary, more complex plant compounds. You know, we know this, we think about this in the context of quality, and we're trying to build protein for quality. It's the same kind of concept. We can also use that line of thinking to also build the inherent immune system of the plant, to build all of these defense chemicals and protective compounds. But again, the plant can't do it if it doesn't have the adequate nutrition in order to facilitate that process. And that's really what it comes down to. And so, you know, there's a lot of discussion now about nutrition-based strategies, and fair enough, a lot of skepticism and mixed opinions and discussions on this, which is, of course, all very fine. It's a newer idea, perhaps, that we are discussing. But I just also wanted to point out to the listeners that, in fact, there's a huge number of studies and research and evidence that do link this discussion of nutrition to heightened immunity and therefore lower pest and disease pressure when we address a nutrition-based strategy. And this is just a very small selection of a handful of papers that explore this topic. And of course, let's be clear, it's not to say that therefore all of these studies and others are always in the positive. I mean, it's a mixed bag of results. But I just really want to emphasize in this discussion on whether or not it's possible, I would say that there's a significant body of evidence that shows that it is possible. That's not the debate we need to be having. The debate we need to be having is, okay, how do we translate those findings out into the field? It's how do we do it? You know, how do we make it practical? And of course, that's always the challenge with, you know, many concepts and many ideas. And if we can't make something practical, well, then it's pointless. But just to emphasize, that is the debate that we need to be having, is how do we apply the knowledge? How do we do it in the field? It's not whether or not it's possible. The evidence is there to support the role of nutrition in supporting plant immunity. It's how do we make it practical? And I just share this slide from, this was a presentation just last week, a week or two ago at the Oxford Real Farming Conference. And I encourage people to log on to YouTube at the end of the month. They'll find this one. It's not up there yet, but this was just one small example. A researcher from the University of Edinburgh looking at specifically nitrogen and the excess supply of nitrogen, which can induce greater herbivore pressure, greater insect pressure, as well as disease. And she's quoting 30 odd papers here in this direction. And the majority of these point to this, that it is too much nitrogen in the system inducing these problems. Now, it can also go the other way. It can also have a deficiency of minerals, nitrogen including. There are some immune pathways in the plant which are nitrogen dependent. So, of course, a deficiency of nitrogen can also limit plant immunity through those pathways as well. So it was a very interesting presentation. I encourage people to look at it, and she equally shared another slide looking at the effects of some of our plant protection products and how they can also induce a more susceptible crop to disease. And she also shared a few studies on this. So again, just pointing out that there's a good body of evidence to support the principle behind this. And really in this context, in the discussion of nitrogen, you know, there's a whole host of things to discuss, but in the example of nitrogen, the point is that the plant, when it is unable to produce more complex and more complete proteins, is the problem. If we have too much free nitrates, too much ammonium, too much smaller amino acids, too much of the building blocks, not enough of the complete and complex proteins, that's where the problems then stem. And so this is just an example illustrating this, is that, again, the classic view of nitrogen, we often, the use of ammonium and nitrate, you know, it dominates our thinking as these are the two primary forms of nitrogen that plants utilize. And it's just to say that it's not just about applying nitrogen. It's not just about having high nitrogen in the crop that is the problem. It's also about all of the other synergistic minerals that make nitrogen work, that are involved in nitrogen metabolism. And so here's an example here. When ammonium enters the plant, it is dependent on manganese and magnesium to convert that ammonium into the amino acid, glutamine, and then onwards from there into proteins. Now, nitrate, on the other hand, when it enters the plant, there are other minerals that are also involved in the conversion of that nitrate also into amino acids. So molybdenum and sulfur are required as part of a nitrate reductase enzyme. Then we also need iron for the nitrite reductase enzyme. And then once again, we need manganese and magnesium. So the point is to say that if you don't have some of these minerals in red, particularly these trace minerals, a lot of them are on the list, you can see, if we don't have adequate supply of these in balance with the nitrogen supply, well, then the plant cannot convert those building blocks into amino acids and then onwards into proteins. It's then about sulfur again, boron, converting those amino acids onwards into proteins. And so that's the broader discussion that we really need to have. And you know, going back to this study here, this was, you know, wasn't emphasized enough in this discussion. It's just linking high nitrogen availability, linking too much simple amino acids as part of the problem. We needed greater discussion on the role of these other synergistic minerals in facilitating the conversion of complete and complex proteins. So really, you know, opening up, we need much more of a discussion about holistic plant nutrition. You know, it's not just about the major minerals, it's not just about the one or any one, it's about how that one works with the others to fulfill its functions and metabolic pathways. So that's the broader conversation that we need to have. And so just a few quick words on the...
Just going to ask you a wee question there. Just if you went back a slide. So where did you get the information to actually influence this? you know, I mean, we know, you know, most people apply manganese. We've seen that in terms of fungicidal response as well. You know, a couple of kilos of manganese in the tank as you go through our 4, possibly 5 timings, if you like. And there is, you know, almost in our crop growing strategies on the winter cereals, we're just about getting to the stage where you're going with the nutrition pass with the sprayer, if you like, on foliar. So is this soil chemistry in here, or is this foliar, or where do you go with that?
Yeah, it's a combination of both. Soil taking a slightly more longer term and bigger picture view of what's happening in terms of your mineral supply, but really on a season to season and a more fine-tuned basis. It's really plant analysis. Let me stress, just because manganese and magnesium are involved in that conversion, it doesn't necessarily mean you have to apply them, but it does mean you need to be looking out for them and addressing them if they are limiting. So really the answer to your question, the most precise way to do it is through a regular and much more fine-tuned plant analysis. And that could be tissue analysis, or sap analysis seems to be a technique that's gaining a lot more traction these days. But, you know, either one, it's about using the leaf analysis in a much more precise and more targeted approach. You would ask the plant directly. Don't make assumptions. Ask the plant directly, find out exactly what the limitations are and address them.
Yeah, which is fantastic to hear as well. And that's ultimately what we're trying to do at the strategic farm. And one of our biggest ones, you know, I see you've got iron there as well. I mean, the toxicity of iron is well and truly documented, you know, in terms of stunted plants, and especially in oilseed rape growth, and usually associated with compaction. What's your view on that sort of correlation?
Yeah, of course, that's very common. I've seen that a lot in Scotland too. So yes, and again, I think that's a good example that emphasizes the point that I also just made. You know, don't apply iron just because it's involved in that pathway. You know, ask the plant directly, and then address those limitations that are actually required. Okay, now if you have a historical knowledge of knowing that iron is regularly and consistently high, or anything else that's regularly or consistently low for that matter, you know, of course you can start to bring in some prophylactic and preventative strategies to address those if you know they're going to be very common problems. But in relation to iron, yeah, of course, dealing with an excess is difficult. It's much harder than dealing with a limitation. If something's low, simply apply it. If something's high, it becomes much more difficult. Now, one of the things you could be looking at there is then also the balance or the ratio of certain minerals. So we know that, for example, iron and again manganese have a relationship. So they operate a bit like a seesaw. So if you were having high iron in the tissue, you could apply. We know that high iron in the plant can block or antagonize manganese in the plant. So I would be looking at manganese and trying to increase the level of manganese to the top of its ideal range, not to push it into excess, but not just to have it in the bottom of the target range, but I would be pushing it to luxury levels of the target range, keep it on the higher end to just try and compensate for that excessive iron. So strategies like that could be used, for example.
And very difficult in that, you know, it's such a short window, isn't it? You know, and the one that we usually get is late season oilseed rape, which generally will see high iron levels, but, you know, who applies manganese to oilseed rape? Well, you know, probably a way to try it, to be perfectly honest, but, you know, it's not historically been the advice we've been given. And it does go back to Eric, is like, we'll get a soil test and it'll tell you all these trace elements. You think, well, is that high? Is it good? Is it bad? I suppose the question you get into is, how do you know?
Yes, so, you know, well, with the tissue analysis, you will have some ideal levels, some guideline levels. If the lab's just providing you with a bunch of numbers, of course, that's rather unuseful. You need a reference point, you need a target range to know. So if your lab's not providing that, I would change labs, would be my suggestion. Now, you have raised, though, another important point, which is going to come up on one of my future slides, which is there is a bit of a question mark over those target ranges. Many of our ideal levels that we currently use, many of them are 30-odd plus years old. They haven't necessarily changed with the updates with varieties, with the increases in yield. There's a big question mark over how reliable our target ranges are, especially from a tissue test point of view. So I acknowledge your question. It is a challenge. It is a difficulty. It is a challenge, and I would agree that there's a lot of knowledge gaps in this discussion around nutritional-based approaches. But I would just, again, I come back to that earlier point. Is like there is a good grounding of evidence to support the concepts. What we do need to be doing, which is exactly what you're doing, and my full credit to AHDB for doing this. We need to be having discussions like this, running trials like this with David. We've invested for decades in pesticide technology, in genetic technology, in fertilizer technology. We've invested heavily in all of these kind of areas. Looking at this specific focus hasn't been a research agenda. So, if we apply some of the brilliant minds in the UK to this, of course, some of these answers can emerge, and in fact they will have to. We know plant protection products are going to be withdrawn. We know we're heading in this direction. So the time is now to have these discussions.
So interesting point you mentioned there about how some of these figures, that reference figures that are being used nowadays are 30 plus years old, and it might be different for modern varieties, modern yield ranges, etc. Are we then better to look at ratios between different nutrients so that it doesn't necessarily matter what the level is, as long as it's balanced correctly with the rest of the nutrition within the plant? Is that maybe a better way to look at it, which might negate the need to start research again and looking and seeing what modern levels need to be?
Yes, no, it's a very good suggestion. There are many examples where using an approach like that can be very beneficial, in exactly as you say. Although we may not know precisely, or there is a question mark, at least we do know certain minerals have relationships. They are synergists or they can be antagonists, and consequently, yes, trying to then optimize some of those ratios can be a useful strategy. Yes, absolutely. I think that's a good point.
Yep, we're just going to go in here with a very quick poll. The questions are going quite wild here, so a very quick poll, Christian, just to get a few points out there, and it's just a question. So, how many people apply manganese to OSR? Just as a question, and I suppose in this context we're meaning the early establishment, to get oilseed rape off the ground at establishment or very soon thereafter. So just a very quick yes or no. Just we've got Gordon and Andy on there as well, which is absolutely right. They're saying different parts of the country, different people have many different ways of doing things. So if we close the poll down there, Christian. We'll just hand back to Joel on that one then, won't we?
Can you see the results there, Joel? Sorry to jump in.
I can.
Okay,
yes, I can.
Sometimes the person sharing the screen can't. So, okay, cool.
Yes, I can. Yep, very interesting. So there you go. And I think that speaks volumes as to, perhaps why many people have a skeptical and mixed opinion about a nutritional approach. Let me stress, it does need to be targeted. We're not going to just guess. We need to use a scientific approach like we do in all other areas of agriculture. We need to apply the same thinking. And so this is just one small example. We're just talking about ammonium and nitrate here. We could have this conversation about many other things too, many other minerals too. And equally, just to stress, let's also not get bogged down in thinking just about nitrate and ammonium. Plants can also absorb amino acids directly from the soil, and even small proteins called peptides. They can absorb those directly from the soil. Plants will also consume bacteria. They will eat bacteria. Bacteria will be engulfed into the root cells as they are subdividing, and the plants will digest and break down bacteria for nutrition. So this is also, again, a much wider conversation that we need to have. Here I am emphasizing this point about, well, you need all these minerals to build amino acids. Well, plants, you can save all of that energy and all of that hassle by not using inorganic minerals in the first place and trying to feed the plant directly amino acids, save its energy, save these metabolic pathways, and feed directly organic fractions of nitrogen, amino acids and peptides, for example. So there's a whole much broader conversation that we could have as well. Okay, so just I threw this in here because I saw it was in the suite of tests that you'll be looking at for David's farm. And just to say that at a farmer level, at a field level, the refractometer is one tool that we can use to measure plant health, to measure photosynthetic potential of the plant. Now, I will stress it's a bit of a crude tool, can be a little blunt. There's a lot of noise in the readings. There's a lot of variables. There's a lot of environmental factors that influence the reading. But essentially what this tool can do is we will extract a bit of plant sap, drop it on and have a look through the meter. And this is what we can see. And what we're really measuring is what's called the total dissolved solids, or really we're measuring the products of photosynthesis, the sugars, the carbohydrates, those proteins, those defense chemicals, all these kinds of things. And I just have to skim over here because we don't, for the essence of time, but we're looking for a higher and as blurry reading as possible. And when we have a higher reading, that simply says we have more products of photosynthesis, more photosynthates, more sugars, carbohydrates, etc. in that plant sap. So this plant on the right has a greater photosynthetic functionality. And if I just come back to this slide, this is what we're measuring with the refractometer. It's the products of photosynthesis. It's all of these things on the right. It's all of those sugars, all of those carbohydrate proteins, defense chemicals. And again, you can't synthesize those if you have a limitation or an imbalance of the essential minerals in gold, in the yellow writing there. If you don't have the mineral, you can't have these complex and secondary compounds, these immune enhancing compounds, and these proteins and qualities, etc. So that's what you're measuring with a refractometer, is these products of photosynthesis, and therefore we are making an assumption about the mineral status based on measuring those products on the right.
Joel,
so just
what?
Yeah, okay, sure.
Sorry. What sort of levels as a guideline? I mean, when we've done bricks here in the past, we don't get very high readings, and you hear of guys in the south getting 20 and 25, and you think, well, do you think we can achieve that level of bricks, or have we got to be more content with 10 to 14? Have you any guidance as to where we should be worried, or do we just keep pushing for higher and higher number?
Yeah, so as I say, there are other environmental factors, and sunny days versus cloudy days is a big obvious one. So this is going to be always a limitation in Scotland. The plant will photosynthesize higher, more active during sunny days. So that's part of your answer. But yes, of course you can always do better. And the general rule of thumb that many would say is you want to get a minimum of 12 and above. Now, but it's different for different crops. So you'd really have to Google that. You'll find some charts for what the minimum and kind of what the ideal levels are for different crops. So for example, potatoes are known to be lower. For example, I think it's 7 for them. You want 7 and above is adequate. So it does depend on the crop, but generally it's this 12 figure. But you touched on the answer already. Just because there's some information out there that says the ideal is this or that, the answer is you should push for as high as you can get it. Just because 12 may be where we see more of these immune supporting properties come through, it's not to say that you shouldn't strive for 14 or 16 or 18. As long as you've got those mineral catalysts there, that's what's going to help drive that process. So as high as possible is the answer. Generally,
how do you do that though? The difficulty we have, so just jumping in, so we've done a bit of bricks, with our business groups and, with the sandals on and whatnot, which is great. Point out a bricks meter, it's 25 quid on Amazon. It's nothing fancy. Just go get one and get out in the field and do it. It's not difficult. But we've seen, as David says, bricks here from 7 to 12, and that's generally our range. Now, you can have bricks up 12, 13, but the minute you apply the nitrogen, bang, it's off the cliff. So to keep it high, how would you keep it high? You know, do you have to stop applying nutrition?
Yeah, and nitrogen is known for being a big culprit here. Again, you're on the right track, but it's not just nitrogen per se. Again, it's that form of nitrogen that you're using, and then not supplying the adequate trace minerals, etc., to convert that. Nitrogen is a building block to build proteins to make your bricks high. You know, it is a raw ingredient to get you there. But it's that form and the unbalanced supply of that, with the traces not keeping up with nitrogen supply. I mean, I would encourage you to use, look, just for example, and I guess it'll come to it in my next summary slide, but just to say that, you know, for just one small example, urea as a foliar is very beneficial in this regard. It's a form of nitrogen that's very energy efficient to be converted into those aminos and proteins, which will lift your bricks. Put urea on the soil, okay, then it gets converted to ammonium and nitrates, okay, less favorable. But put urea directly onto the foliage, the urea molecule is absorbed as urea, and that is a form that's more beneficial. So I'd be looking at, for example, things like foliar urea, but yes, I would be looking at strategies to cut down my nitrogen over time, and perhaps I'll cut to my next slide, because this is kind of the next slide anyway, about, okay, how do we take some steps forward? How do we begin a process of transition? And just a few kind of closing thoughts here. One, okay, we'll start simple, you know, soil health principles, you know, adopt those 5 soil health principles as best as possible or as often as possible. You know, I think that's a very, very good place to start. But then these next couple link to your question, Chris, you know, so I would then also use a strategy of carbon-based inputs, and that simply means always putting a carbon source down with any time you're putting a fertilizer down. And this is often discussed in a context of integrated nutrient management, combining organic with inorganic. And so, for example, yes, use FYM and compost with your synthetics. Then you're putting a carbon base, a carbon structure there to hold on to that nitrogen and slow the release and provide some of those organic fractions of nitrogen, the amino acids, etc., which are more supportive of plant health. Now, in a liquid context, that could be things like humic or fulvic acids, even molasses, kelp, fish hydrolysates, these kinds of things. These are all carbon sources that you could mix with liquid nutrients in a foliar or, again, soil applied. Again, I mentioned before, foliar inputs, I would really encourage people to look at this as where you can use less fertilizer and be more targeted, be more efficient. But again, you want to use your leaf analysis to steer that process. Again, I mentioned the example of urea and carbon-based urea as well. Using carbon sources with that urea, also very beneficial. And again, supplying the trace minerals to support the conversion. Improving soil biology, that's of course at the top of everyone's list these days. You know, in a way, such a very simple thing to improve. Just keep the soil covered with living plants. If you've got living plants, living roots and root exudates, you are providing the best food source for supporting soil biology that you can. So, you know, again, coming back to those soil health principles there. And then kind of just bringing in one last part of a discussion on, okay, cutting down pesticides, at least, you know, bringing in more ecological thinking, you know, ecological intensification. So you touched on that already as well today, so I won't dwell, but, you know, again, wildlife friendly field margins, using more diversity of rotations, but also in-field diversity. I do think there's great potential in intercropping. And so with that diversity, this brings a lot greater multifunctionality. And there's a whole suite of evidence that shows that if you've got more diversity in the system or even around the field margins, across the farm scale, into the field scale, all of those things help lower the disease pressure, lower the insect pressure. And again, there's very good evidence, not to say that you'll completely control them, but they will lower the pressure. And there's very good evidence to support that as part of integrated pest management strategies. So yeah, in conclusion, all I was going to say was encourage people to not just think about their soils in the way of 3 on the left here in physics, chemistry, and biology. I would say we have to bring in a 4th part of our soil health thinking, which is the role of plants. Plants have to sit in there, and that could be plant diversity in what, like the context I was just talking about there, or the role of plant nutrition? Do we have the minerals in optimizing plant health? Because when we optimize plant health at the top there, well, that also influences the chemistry, the physics, and the biology. They all feed and interlink together. So that would be my closing thought. We can have some discussion now.
Wow. Yeah, certainly a lot of ground covered there. And, you know, apologies to those. We did extend the time of the webinar just because we knew we were never going to do it in an hour. So we did extend it to 12, you know, so we've a bit of time there as well. So I'd ask all our panelists to turn on their cameras now as well, and we're going to open up to a few questions as well. So there's obviously quite a bit going on there in terms of crops and definitions. So Joel, just a few for you. What's your thought on going from variable rate nitrogen to variable rate magnesium, as chlorophyll is a block of magnesium with a nitrogen at each corner? I have done this area regarding crop greening effect can be seen within days. For comment, if you understand that.
Yeah, I mean, I think it's, again, that's a good example of also what I was discussing before. We need to think about all of the nutrition, not just one variable in isolation. And magnesium is another example. You need to supply magnesium with nitrogen to build chlorophyll. They form part of that chlorophyll molecule. So that's also a common problem. We see not enough magnesium in the plant. And also, this is very common in high magnesium soils, which is very counterintuitive, but it's very common in high magnesium plants growing in high magnesium soils to be magnesium deficient. They seem to struggle to get it out of the soil. So in that sense, supplying magnesium alongside nitrogen helps to build better chlorophyll and therefore drive photosynthetic potential much better. So yes, that's another good example. And in terms of variable rate, I mean, sure, I know that's a lot of people are moving down that pathway these days, and we need greater precision approaches. I don't have any particular objection to that. Of course, if there's a lot of field variability with magnesium, for example, then sure, you could address that accordingly. But I would still stress you've really got to come back to that tissue analysis. Just because the mineral is or isn't in the soil, it doesn't mean that it is or isn't coming up into the plant. So I would stress, I think, if you really want to take it to the next level, it's during in season, you've really got to be using more of a tissue analysis to then, yeah, really fine tune that decision.
Yeah, and where are we going here? You know, we've got urease and inhibitors, which, you know, have been played around with, and some have had disasters and some have had probably better results. But, you know, probably going forward in the next 10, 15 years, you know, we're just not going to be allowed to throw pellets of fertilizer about, you know, out of the back of a ferret spreader. So what about this urease inhibitors? Is that good news, bad news? What's the thoughts on that? Open up. We'll go with Joel first and then Eric. Fiona after that.
Yeah, I think there's two sides. I have mixed feelings, two sides to that. Of course, it's good. Anything we can do to limit nitrogen escape into the system is we need to be putting on the table. Of course, my just slight reservation is, you know, it's not changing. We're not addressing the habit. You know, we're providing a tool to maintain the habit of nitrogen fertilization. I think we also need a little bit of mindsets and thinking about how we manage that nitrogen better. And so, you know, from that point of view, I guess I have some point. And I think the other concern is we do know that the inhibitors do enter the plant, where they also inhibit the urease enzyme within plant tissues. And the plant needs urease enzyme to convert urea into amino acids and proteins. And so we also know that it can block that conversion within plants. So, which is then a point to say that, well, then that urea gets stuck in the plant as urea. The plant doesn't want urea, it wants amino acids and proteins. It wants those organic fractions. So we can also block that conversion, which then leads to the problems we were talking about of making the plant more susceptible to disease as well. So, you know, so I have some reservations in that sense, but of course, I know that they do help to prevent nitrogen escape into the ecosystem, and that is something we also need to be hugely putting on the agenda.
Yeah, absolutely. And an interesting, you know, protocol there to actually with these urease inhibitors is actually to measure disease prevalence with them as opposed to a normal strategy, which I hadn't really thought about as well. Eric, have you comments?
Yeah, I mean, you've got to differentiate between nitrification and urease inhibitors. Nitrification inhibitors reduce emissions of nitrous oxide from the soil and decrease leaching of soil nitrate. There are two products probably most widely available, DCD, dicydiamide, and DMPP. Those are most commonly available nitrification inhibitors. We know, just picking up on the comment that Joel made there, we know that for nitrification inhibitors that clover and lettuce, there are known toxic reactions in using nitrification inhibitors in these two crops. So we need to be careful as to what we're doing there. It's not a universal solution. For urease inhibitors are used to decrease emissions of ammonia, the NH3, following hydrolysis of urea in the soil or on the soil. And both nitrification and urease inhibitors are available. There is limited evidence of them being effective in Scotland, but they're not widely used at present due to the poor cost effectiveness under conventional economic analysis at the farm gate. For example, the analysis that has historically been conducted hasn't considered the externalities of the environment or societal costs of leaching of nitrate into water.
Yeah.
I think we also need to note that nitrification inhibitors have also been found in water runoff, and they can also be a contaminant in water streams. So it's not a clear-cut case just now as to, yes, we should be using them. I think the industry and government must carefully assess the benefits and any negative aspects of both nitrification and urease inhibitors. There has been a consultation out in England that was asking about urease inhibitors for urea to be made mandatory. That was only in England. The consultation did not go out in Scotland, and the DEFRA consultation made no comment on liquid fertilizer products. Now, most liquids do have a proportion of urea in the recipe. So it'd either be urea plus ammonium nitrate or urea plus ammonium nitrate plus ammonium sulfate as a liquid fertilizer. So yeah, big question marks whether urease inhibitors and or nitrification inhibitors should be utilized or not.
An interesting aspect I hadn't really thought about was, the actual inhibitor itself being a pollutant as well, which, is an interesting concept. I hadn't entered my mind or my capacity, if you like, as well. Fiona, where are you on that with all the trials you've got to do this year as well? It's quite a lot of information there to bombard us.
No, there's lots that we could include in terms of the treatments we test, but I would go back to a comment that Joel just made about where inhibitors sit in the context of what we're doing. So if you look at the marginal abatement cost curves that sit around where we might get the big wins for going to net zero, at the top it's the increased use of grain legumes. Second is the more precise targeted way of doing that, so variable rate approaches, and then you've got intercropping, and then you have inhibitors as having, sure, they've got potential to improve the situation, but they come further down. So you've done everything else to reduce and target your nitrogen before you get to the point you're thinking about those. So I think absolutely they have a place, but there's a cost, both monetary and in terms of the effect they have on the plant and the effect that they have in turn as pollutants. So I think it's just that idea that they sit somewhere along our decision tree, but absolutely not at the top of it.
Fantastic. Now look, we have a couple of questions here, and we'll probably start off with seed treatments, and by that I'm talking about actual nutritional seed treatments. So we can inoculate seed, we can apply fiberphos to seed, we've got manganese, we've got zinc, we've got all these things. Where are we with that train of thought? Or part of me is thinking you can't possibly get enough seed treatment onto seed to give it any benefit at all. But is that the case? Has anyone done studies? If we go back around the same order, Joel, what's your thoughts?
Yeah, you're exactly right. I mean, they can be very useful. You know, they're just to help that early establishment phase and get things going. Of course, you can't supply enough for the full crop demands, but they can be they've proven to be very useful. For example, in highly calcareous soils with particularly trace minerals, you know, which are low in availability. So examples like that. So yes, it's not really designed to supply the plant with a full crop year supply. It's about just helping that establishment and that pop-up phase. I would suggest that I think there's a lot more gains to be had. However, looking at more of the biologicals and biostimulants, and then having the nutrients as a background component of a seed treatment mix. It's the biologicals and the biostimulants that seem to trigger much more of that earlier rooting, much greater root branching, and some of those favorable root traits. You achieve that more from some of those other stimulants, and the nutrition being more of a background part of that.
Yep. Okay. Interesting. Eric, we've obviously done quite a bit of this in the past with yourself. What's your thoughts?
The cultivation and drilling technique have interactions with this, because your soil quality, the consolidation of your seed bed will ultimately reflect in, particularly with manganese, in your availability of manganese to the young crop. We have seen short-term responses to manganese being applied as a seed treatment. That can be useful under extreme circumstances, because you can't apply manganese clearly to the soil. It's either going to go on as a seed treatment or applied foliarly, because manganese is a trivalent. You get Mn plus and Mn2 plus, and the valency in the soil will reflect both temperature and soil moisture. So until the crop is up to 2 leaves, it's very difficult to get any augmentation in from foliar manganese. So to getting a crop through to that stage, it can be useful. We do also need to pay attention to phosphate nutrition. Phosphate is very, very important for establishing crops and getting root growth in that initial crop development phase. So both have a place. Joe made comment on biologicals and biostimulants, and the work that we've done there within Scottish Agronomy on those 2 products. It's very, very difficult to have control over the environment to ensure there are conducive conditions for the biostimulants to actually work. We've done a lot of work within a project I'm involved with on arbuscular mycorrhizal fungi, and we know that certain crops, AMF will not establish on the roots at all, and even on those conducive crops, the inoculation of the roots is being suppressed by the high phosphate status nature of the soils in which we're growing these crops in. So there's a lot yet to go.
Yeah, a lot we need to understand as well. Yeah, Fiona, you'll see a lot in your crop doctor tours as well, I'm guessing.
Yeah, no, and I would probably go back over comments that both Eric and Joel have made, but there are actually probably very few examples of seed treatments where there isn't some crossover between the nutritional benefit. So if you take something like phosphates, that they also have an effect on switching on elicitor responses as well as a nutrient piece. And something like manganese is so fundamental to so many metabolic pathways that there's an interplay. Our own research, I mean, we're in a much better position to look at what genes are being upregulated in plants. So where they are switching on those responses, our research has been around, as you would imagine, their influence on ramularia. But just to flag again, there's a negative side to switching on induced resistant or elicitor responses where the plant's not needing them. So again, it's coming back to that idea of a targeted approach to doing this.
And that will be coming from presumably our soil tests, or, well, it has to be soil tests if we're going seed route. So we would then be looking at the soil test in depth and actually come up with a seed treatment, if you like, of these trace elements, nutrition, to try and benefit the seed in that field in that scenario.
Yeah, absolutely. It's putting it together. And I agree, again, Joel made a comment that the concepts around this are proven, but actually in terms of the current Scottish situation for our major pathogens, we are still in a big knowledge gap.
Yep, I absolutely, completely agree with that. So we move on then to looking at leaf tissue tests. So when and how early? So if we're looking at wheat crops, we've obviously got this trial that we're doing at Balbirnie with David, and 15th of February is our open period, which rightly or wrongly, should we be applying nitrogen? We're going to have bricks tests, we're going to have tissue tests. Is that now, Joel? We'll open that up first. How often do you do tissue tests? When? And it's back to the question of influence in bricks, I suppose, isn't it? And David, feel free to come in on that as well. Obviously, you've done bricks a long time.
Yeah, it's, you've, like all things, there's that middle ground of what's theoretically possible and what's practical. So you've got to align it to what's feasible. I think I would say that the point of taking an analysis is to react on it. So I wouldn't necessarily take it for the sake of taking it if you knew you were then going to go out with a foliar pass, for example, and the timings of that work. For example, of course, many people would link this then with their fungicide passes. I would be working back from there to say, okay, well, I'm going out anyway. What can I address while I'm going out? That's kind of one way to think about it. But equally, it really comes down to the economics. I mean, in intensive, for example, veg scenarios, people will be doing, an analysis sometimes weekly, but perhaps more maybe every 2 or 3 weeks, something like that. So, depending on how intense and how serious you want to get about the approach, it could be as frequently as every couple of weeks. But otherwise, choose your key times when you're going out anyway and address accordingly.
Yeah, good. And I suppose from my side of it, I've still got to get semi-clear in your head. It's like, it's nitrogen, the little and often approach, the what nitrogen, the foliar, is it foliar first, keep the bricks high early? What are you, there's so many questions here. What are you trying to achieve with it? Do you want to keep the bricks up? Is that the key goal, or do you let it dip early season? And I suppose that lets the disease in, doesn't it? I don't know.
It depends on that one. I mean, yeah, the broad goal is sure, keep it up. I mean, and monitor your bricks regularly. If you see a severe drop, I think that's the point. I mentioned that the bricks can be a bit of a blunt tool, and it is. But I think where it gains a little more value is when you're doing more regular assessments. It's the trends and the patterns, like a lot of things. It's the trends and patterns over time that then can give you more meaning. So if you see a significant drop all of a sudden, then I would be trying to take an action to address that. And yes, the general approach with nitrogen, again, it's just about what's practical, but the approach is definitely little and often. The problem is we put large doses of nitrogen on in one hit, and I know we do that for very practical reasons. I think that is the problem. It's maybe not necessarily the quantity of nitrogen that you're always applying, it's that big dose at once that overwhelms the system. So I would support a little and often approach, but I know practically that becomes difficult. Then there are strategies that people, for example, use is to cut their soil applied nitrogen down and then substitute up with the foliars. So, for example, cut nitrogen down by 50%, but then substitute up with more regular foliars. So, and then when I say substituting up into the foliar, you're not putting that full other 50% off. You're going to gain some efficiencies through that foliar pathway, and so that's where you can make that gain. So then those are kind of, for example, strategies. I know others that just do more of a slow and steady approach, trim it down by 10% per year, and again always combine with carbon to help stabilize. Those are kind of some examples of what some people could do.
Yeah, and a very interesting point there with it as well, and we'll come to Eric and Fiona as well. Now, one of the questions that we've got in, regarding the Mulder's chart of soil interaction, does that hold true in your eyes, for plant nutrient interactions? Do you think that's the way we should be looking?
Yes, I mean that concept. Yes, the Mulder's chart was derived specifically for the soil, and so the same interactions don't always apply in the leaf, but there are many overlaps, and there are other interactions. That comes to David's question earlier about the balance between minerals. So yes, I think that kind of a chart for plant health and plant analysis is a similar and relevant strategy. I do think that the balance between minerals is a very important thing.
Yeah, absolutely. And I'll just add this in when we get to Eric and Fiona. Do you think we're actually teaching this at college level enough? Was one that was proposed there. So, you know, are we teaching the young budding agronomists and farmers coming through with SAC, SRUC, and Eric, you do a bit of this as well. You know, are we pushing this hard enough with them at it? You know, so we'll hand over to Eric there quite a bit to try and answer and get through.
Okay, I'll take that question first, and I'll flip back to the nutrition. No, I don't think we're teaching the basics at an education level, but we're not joining up all the dots or considering all the linkages that are there. In reality, biology, and we're talking about biology here, is very, very complex, and there's lots of interactions, very complex interactions. Joel was talking about nitrogen. I'll just complement that by talking on a slightly different aspect of nutrition and talk about phosphate. Soil-derived phosphate provides, on average, about 80% of crop phosphate requirements. It can range between 42% and 100%, but average phosphate recovery from broadcast triple superphosphate, for example, is only about 4%. And often applying fresh phosphate doesn't overcome efficiencies. If the roots can't get the supply from the soil, then should phosphate and sulfates be supplemented by foliar application for higher yielding crops? That's a throwback question. But you know, rather than just looking at leaf tissue analysis or sap analysis, there's useful evidence coming from likes of Roger Sylvester Bradley and ADAS that monitoring grain phosphate levels could provide complementary nutritional monitoring technique, particularly as it provides information on whether a crop's got enough nutrient through the season. I appreciate, Chris, from your perspective, it's not an interaction or a closed loop that you can actually engage on within a season, but it then informs your next season's growth. And evidence suggests that the grain phosphate levels of around 3,200 milligrams per kilogram is a good critical threshold to have in mind, as yield does tend to stop rising above that level. But you know, using tissue analysis, yes, has its place. I think introducing here the concept of using grain analysis at harvest time as a feedback mechanism informing your nutritional choice on a farm is I think important.
Good, thank you. And Fiona, to yourself.
Well, I suppose I wanted to pick up again on the education element. I mean, absolutely, the sort of increasing content around the soil chemistry and the interplays and the precision ag idea are increasingly part of courses. It would depend what entry level you were going in at and where it sits. But I also wanted to add that it's a continuing part of all our development, so that the idea that the CPD that practicing farmers use, so that, you know, and advisors, so sitting around particularly the facts course, again, you know, the inclusion of this type of information there is a key feature. Chris, is it worth me saying a little bit more about how the trial will be designed at the home farm?
Yeah, absolutely, Fiona. We've a wee bit of time, just.
Yeah, we kind of moved over that quite rapidly for time reasons in the slides, but what we're doing is a tramline trial with 3 replications where we still to decide the treatments that will actually go on. But the idea being that as an untreated control, a standard farm practice nutrition tramlines, and then the real-time reactive element where we're using these new techniques to then try and input what we're doing. So I think the kind of debate now about whether we include the, for example, the foliar ureas, you know, is something that we could perhaps explore.
Yep, I absolutely agree, Fiona. Yeah. And it's great, you know, it is a live trial that we are hosting, and it's almost, you know, it's not Fiona against us, but it kind of ingests that's the way it's going to be. It's, you know, these are trying to get us ideas to try and push it as far as we can in terms of crop husbandry. I'm sure you'd agree, David, just before we close.
Yeah. The basis of the trial has been set out, but what actually happens is you know, we'll decide that as we go through the season. And I'm fascinated by the foliar urea, and Eric's heard me talk about this before, as have many of his colleagues. And I find it fascinating that historical data appears to show there's no benefit, and yet there seems to be plenty information out there that suggests we can get some real benefits from it, in terms of nitrogen use efficiency potentially and disease reduction within the plant. So I'm keen to see if we can make some mileage on that, because you know, if we can start knocking 20-25% off our nitrogen usage, then for many reasons, financial, environmental, policy, you know, we're going to start making big inroads into some of the goals that we've got in front of us as an industry.
Absolutely, and I think it does come down to investment versus effectiveness, and we will be using Farm Bench to quantify everything we do, so we'll know, you know, if we have broken the budget completely, or if we're ahead of budget. And you know, ultimately, you've got to bear in mind that you know, we are potentially, depending on what happens after Brexit, still still facing you know, a loss of chemistry coming towards us. So that is very difficult. Now, look, I'll apologise. There's a huge amount of questions come in there, and we're just not going to get through them all. And we will answer them as a group, and they'll be posted on the AHDB website. And I'm sure Faz can have this as well, just in case, whichever comes first. So I'd just like to say thanks very much, especially to Fiona and Eric for joining us, and David, the host, and Joel, who got up at 5 o'clock this morning to join us from Canada. So look, thanks very much to you guys there. And you know, so much information, it's difficult to process, but you know, no doubt we will be doing another webinar fairly soon. So thanks very much, everyone, for sticking with us, and we'll catch up with you soon.